I am very happy to introduce Professor Arvind Rao from Michigan Medical School. He is a professor in the Department of Computational Medicine and Bioinformatics at the University of Michigan. And his group actually uses image analysis and machine learning methods to link. Yes, madam. Okay, okay, okay. Then I will wait. So his group uses image analysis and machine learning methods to link image-derived phenotypes with genetic data across biological scale, that is single cell tissues and radiology data. And such methods have found application in radiogenomics, drug reprocessing based on phenotype screens and spatial profiling in tissues, as well as in spatial transcriptomics. Professor Arvind received his PhD in electrical engineering and bioinformatics from University of Michigan, specializing in transcriptional genomics, and he was a Lane postdoctoral fellow at CMU, specializing in bioimage informatics. He is also a fellow of American Medical Informatics Association, the Royal College of Pathology in the UK, and American Association of Advancement in Sciences. So he has vast experience in handling biomedical data and applying signal processing, AI, machine learning techniques in cancer research. So I came to a contact with him while discussing one possibility of using geoinformatics and GIS application in cancer research. And that's how we started communicating. And we are very fortunate that he was visiting India and then he could find some time to visit IIST and share his valuable expertise in different areas. This talk is a colloquium talk, so I think many people will get benefited from his expertise. So I invite you, Professor Arvind, for the lecture. Thank you.
Thank you so much. I really appreciate it. So I tend to walk a little bit while talking, but I'll try my best to keep my legs in the same place. Just thought I'll take some time to talk about a variety of different topics, which sort of bring concepts from signal processing, AI, for different aspects of biomedical data science. And I completely understand. I'm standing here in. Will it be feedback if I come too close? Thank you. Oh, thank you. All right. So last year was an interesting year. I understand I'm talking about biomedical science to a very diverse audience today, and it's a little bit cherry picking by design. It's meant to be like a sort of walk through a garden, not meant to get too close, too much of detail. But if we do get up into detail, I'm happy to discuss those things as well. But my hope is I'll cover a slew of different concepts. So would you all mind very much if I walk a little bit? Okay. All right. I might even come down there because I want to prevent feedback with my friendly microphone there. Awesome. Okay. Thank you. Perfect. So I can have a random walk on this garden. All right, great. So last year was an interesting year, right? 2024. I mean, just to give you context, we just had a Nobel Prize handed out a couple of weeks ago. So this year's anniversary has already happened. You had some great Nobel Prizes offered. Last year, something interesting happened. We had 2 Nobel Prizes given to physics and chemistry, both of which went to AI. Something very interesting and subtle shifted, right? The message that was being sent out was, for whatever reason, you don't have to even be a physicist to get a Nobel Prize in physics. You don't have to even be a chemist to get a Nobel Prize in chemistry. What does that mean? And then AI was the front and center in that conversation. So what does that even mean? And there are all these conversations that are happening on the sidelines. You're reading about this in newspapers, right? Oh, AI is going to take away all our jobs. You don't need AI. I mean, we don't need human beings anymore. You can sit at home and just get a standard income and AI will do all the work for you. All these parallel narratives that are happening. So where does that leave us? And so the physics one was to Hopfield and Hinton. To be fair, Hopfield had a physics degree, right? But both of them got it for neural networks, which is the fundamental unit for AI. Again, I will use the word AI extremely liberally. I actually don't know what the word AI means. I use it because it's fashionable, right? I'm still struggling to find my own calibration with what I would like to call AI. In the strictest of strict words, I don't think we're anywhere close to AI, but I use it because that's a fairly well-encompassing word. So don't take too much into the word that I'm using. It's just a placeholder like any other word. And with chemistry, the same thing happened. We had Nobel Prize in chemistry given out to Hassabis, Baker, and Jumper. Baker is the only card-carrying chemist in that sense. He's actually a biochemist, biophysicist. Hassabis and Jumper are working for Google DeepMind. They're both computer scientists. And so they shared Nobel Prize for chemistry because they were able to try to use AI to understand protein structure. And then a few years ago, strangely enough, there was Hiroki Katano, who happens to be the CEO of Sony Corporation, right? I know there's a very interesting mix of audience here. To some people, Sony is like, like Facebook. Everybody knows Sony. There's another group in this exact same room that doesn't even know what Sony is. And so for the people who don't know what Sony is, Sony used to be a company that used to make very nice VCRs and tape recorders and Walkmans and those kinds of things. They were really big in technology. And so the former CEO of Sony, Hiroki Katano, said, well, by 2050, we're going to have a very interesting situation. We're going to have a Nobel Prize winning discovery made purely by an AI engine. At least in the first two cases, there were humans involved, right? In this, now what they're saying is you don't even need humans involved. And a robotic AI system will collaborate to create a Nobel Prize winning discovery. What does that mean, right? I wanted to think about it. And 2050 is not that far away, right? For people who think 2050 is really far away, here's a little bit of a wake-up call, right? This conjecture was made about the Nobel Turing Challenge, was made around early 2020, right? Around that time. But in 2022 November is when ChatGPT came out. And suddenly things that we thought could not be done in 50 years have become feasible. And so when I say 2050, it is now looking more like 2030. So what does 2030 mean? Again, to calibrate everybody who's in this room, your batch of juniors next year who are going to start college next year, when they graduate, this will have become a reality. Just to put it in perspective, it's not that far away. So, and of course, companies are selling this kind of narrative very, very royally. They have AI co-scientists being proposed by companies like Google, who got AI co-scientists, and they've already claimed that they can solve some really important problems. Stanford hasn't fallen behind. They've got their own thing. Company called Sakana, the whole bunch of papers that talk about the process of AI-driven science, right? And here's the world people are envisaging, right? Which is that you're going to have a completely AI-driven enterprise. So this is your pharma company of tomorrow, not of day after tomorrow, not of 2 years later, but of tomorrow, where you will have, this kind of robotic instrumentation doing experiments for you 24/7, 365. And this one's going to be a, AI system, in this case, operating with some really fancy machinery that basically will provide hypotheses for your robotic instrumentation to execute. So you're going to have a situation where you will have AI engines taking all the data from the internet, making hypotheses, feeding it to a robotic platform, which then does its experiments, feeds the experimental result back to the AI engine. That update keeps going until you can converge to something. That's a scientific method. But this is a high throughput way of doing the scientific method. All this is happening as we speak, right? So one very important ingredient is missing, as you can imagine, it's the human, right? So let's think about that. So where are we? So let's start from the beginning, and I did promise to make this talk a little bit about AI in drug discovery. So I'm going to spend the first part of the talk discussing AI in drug discovery very generally, and then talk about tributaries to this conversation that will get into more modern topics. So this is a topic where, again, I completely understand where I am located, and I'm very fortunate to be here, by the way. Thank you for inviting me and this great opportunity. But I completely understand that this may be out of the thought space of a lot of individuals. So I'm going to try to orient you step by step. We are studying a disease called triple negative breast cancer. Triple negative breast cancer is triple negative because it's negative for 3 receptors: ER, PR, HER2. ER is estrogen, PR is progesterone, and HER2 is another receptor that's found on cancer cells. For those people who think that breast cancer is only a female problem, think again. Male breast cancer is very much a reality, thanks to all the food that we eat. So don't think you're spared just because you're male, right? So triple negative breast cancer is a thing, and its incidence is going up in Asia. Okay, why it's happening is a different conversation. So bottom line, you now have these highly aggressive cancer cells that are growing that need to be arrested in some way. And nobody knows, because it's a very hard disease to treat, nobody knows what exact combination of drugs will solve this problem. Now, I'm going to use triple negative breast cancer as an example, but you can think of pancreatic cancer, you can think of brain disease. A lot of different cancers exhibit the same properties I will discuss with you today. And the process of screening for drugs is also fairly consistent. So there's going to be an AI twist to this very, very quickly. So what do? We take plates that look a little bit like this. It's not even this thick actually, it's much thinner. But what you have is a plate like this. You have little wells that are little holes that go into these things, in cross section. And what you can now do is in these holes, in these wells, you can now start putting in cells. So you can grow cancer cells in these plates. Now, what's the point of growing so many cancer cells in so many plates? That you can actually screen for different kinds of drugs in a combinatorial sort of manner. So now I can have a robotic platform that goes and checks every single well, pipettes a certain amount of drug, and now I can see what effect that drug has on the cells in that particular well. Okay? And the picture you're seeing is basically a representation of that. So what you're seeing here is these blue circles are the wells where you actually have drugs going in. These other non-blue circles are the positive and negative controls, which is what you need to calibrate your data. Okay. And first things first, this data is messy by definition. This university and your kind of work is very familiar with the nuances of real data problems, right? Satellite data, remote sensing data is never clean. You have to do a lot of pre-processing before you can even get to a semblance of making sense of it. So that is not news to any of you. But bottom line is that these cells, they have very interesting property. Because they grow like crazy, these cells look like open fingers, right? So they have this amoeboid structure which helps them to move. These finger-like protrusions help the cells to move. It's called a mesenchymal phenotype for those that are interested. And that's a general state of these cells. The reason this is important is because once a cancer cell takes seed in the tissue, it can then very quickly move, go to other places, and create what is called metastasis, right? The intent of this screening experiment is to find a set of drugs using AI that will cause a recalcitrance of this finger-like protrusions to something that looks like this. Why does that help? Because when you don't have protrusions, this thing can't move as easily. That's called epithelial state. Okay, so the idea is we want drugs that can change the state of the cell from what looks like this to looks like this. Now you can ask the question, why bother? Just kill the cell. Why are you trying to massage it into something? Just kill the cell. The problem with cancer drugs is they're not very selective, right? So you give somebody, prescribe a cancer drug, it'll hurt the host a lot more than it hurts the cancer cells. So they have all these toxicities that they will experience. It's a horrible, horrible quality of life. And so we obviously want to find variations that will make this process a little bit better for the patient, right? So the intent is to find drugs that have this change in property. So far, so good?
How much time it will take to make that change?
In terms of once you administer the drug?
Yeah.
Okay, so the typical cycle would be in the order of a few weeks. And so this is the pipeline. I'll show you the data. I'll show you the pipeline too. But bottom line, you have all these images, right? So I just showed you this high throughput screening experiment. And why do, just to pass my words a little bit, why is it called high throughput? Because now I can do a lot of drugs simultaneously in a combinatorial fashion, all with a robotic handle, right? So I can do a lot of drugs. That's the combination in combinatorial. And for every such well where I've made a perturbation, I can take a picture. Not just one picture. In fact, I can take 6 pictures, because they are, you want to take them at different resolutions, different, and you know this stuff, right? Remote sensing, hyperspectral. The reason why you take all these things at different resolutions, because information at every scale. So take these images. Now you've got 20,000 images, right? And the task is to find one small subset in this 20,000 image pool where you have images that went typically from this to some images that have this. Good luck trying to find someone to do that for you. You need automation, you need computation. So the natural place to look is machine learning, right? So what does that mean for machine learning? We have a standard pipeline. It does some basic analysis. It segments out the entities that you care about, does some morphology extraction, runs a data mining pipeline. Long story short, you do a little bit of pre-processing. I'm just going to speed through this because this is superficial at one level. But you build a model, and the model looks like this is a standard machine learning model. This is not deep learning. This is a basic random forest model. People that don't come from machine learning, random forest is basically like it sounds. It's a ensemble of trees, decision trees, where every tree makes a vote on what's happening on an image. There are some spots in the front if you'd like. So just like you would take a vote across multiple experts, the same thing you take a vote across multiple models. So it's called an ensemble of models. And what you're getting is a majority vote that tells you the image that I'm being presented, is it this or this? It's a classification problem, right? So when you do this, go through one loop, right? What do you get? We do, after you sift through all of that stuff, you converge to about 350 genes, approximately, give or take 350 drugs that we think could be potent at being able to convert this to this. Okay? But if you go to a clinician and you tell them, hey, I have 350 drugs that I want to test on patients, they will immediately give up, right? Because it's too many drugs to test in a clinical trial. They'll say, if you give me 2, 3, maybe 5, I'm happy to work with that. 350 doesn't work. So we have to figure out ways to prune this set even further. How would we do that? So when Naoto, who's my clinical collaborator, and I talked to him, he said, the results look good, Arvind, but how can we make it better? I said, at the time I'm still very naive, so I said, I need more data. The standard thing, right? Any machine learning guy that's exactly what they'll say. Give me more data. The easiest thing to say. But here's what happened. He then asks me, well, Arvind, that's all very nice, but what data would be most meaningful to your problem? Just saying, give me more data is not helpful, right? What kind of data? And I didn't know what to do. So fortunately, I fell back on something that I think we all use quite a bit, right? One of these platforms, right? Anybody who doesn't use any of these platforms, I'd be interested to see. Just a poll. Okay, so everybody has dealt with at least one such platform. Amazon Prime, Netflix, Facebook, right? At least Blinkit, right? So everybody has done something. Why do I mention that? Because once you've cultivated a history on these platforms will help prioritize items that they know you will come back for. If I like to watch comedy movies from the 80s, if that's my preference. In the beginning, when I started account on Netflix, right, I get all random movies, right? I get some good movies, some movies that I don't like to watch. Very quickly, in about a few cycles, Netflix converges with the fact that I like 80s comedies, right? And then once it's figured me out, it just gives me more of the same. Sometimes a little bit of variation, but more or less it's figured me out. How does it do that? Here's how it does that, right? It's called, there are multiple names for it. There's one called collaborative filtering, recommendation engines, right? Intelligent sampling, if you like to use that word. I'm sure you've seen, and the machine learning construct for it is called active learning, which you've probably seen in some way or form in your own work, right? So one aspect of today's conversation is to tell you that biomedical data and data that you are dealing with is not that different. Data is data. There are some nuances, of course. There's domain-specific aspects, of course. But I think there's a lot to be gained by cross-pollination between disciplines. So I want to keep coming back to that. So the idea behind active learning is the following. You build a training data set like so. Remember the few images I had to begin with? I built my model, and then I use that model and look at all the unseen data. So let's say I had about 33 images as examples to learn from. My total superset is 20,000. So 20,000 minus 33, whatever that number is, right? Those are that number in this unlabeled pool. What the model does, the model, not the human, what the model does is looks at every instance in that unlabeled pool and tries to flag them by the degree of confusion that that image presents. It's called entropy, right? Uncertainty. So you're quantifying uncertainty on a sample and you're presenting that as a way to flag the most meaningful cases to a human for curation. This is the active part of the active learning. And it's actually called entropy-based sampling or uncertainty-based sampling. So you do that, you then run this all the way through to the human in the loop is simply responsible for curating the data. And then once you get more data, you feed it back into the big pool and you build a better model. So you keep going in cycles, just like Netflix, and eventually you now start converging to better and better performance once this thing moves. So now I went from 351 to 256. I can keep going. This is only one round, right? I can keep going. And in the end, I converged to a set of drugs that I think could be worthwhile to try, at least in laboratory, before it goes to clinical trial. Now, going to clinical trial is serious business. Please, let's not underestimate. It's not just finding patients and giving them any which drug that you want, right? It's a very protocol process. But as a way to be able to do that, you need some method of systematically figuring out which ones are going to truly be worth taking forward. So just to now bring some background back to this group, triple negative breast cancer, like I said, is ER/PR HER2 negative, right? And has very poor prognosis. You won't wish it on your worst enemy, right? And then it is extremely heterogeneous in that there is a lot of genetic variability, making it very hard to pinpoint and treat. And so finding drugs for this disease is really, really critical. And that's why we are solving this, trying to work on this problem. So long story short, I told you I found a bunch of genes, and I told you I came to about 356 genes, and 256 genes kept reducing the set. I keep saying genes just to remind you from, maybe your 10th, maybe 11th standard, right? You did encounter something called DNA, right? DNA is organized in genes. And so essentially genes are those regions of the DNA that code for proteins, things that actually do the work of the cell, right? And so what you're seeing here is a bunch of proteins or genes which were now implicated by this purely data-driven pipeline. I want to emphasize that. This was discovered by a purely data-driven pipeline. The only role a human had was to say if an image was this or this. That's all. There was no biological knowledge injected into this process. And we discovered these genes. I was obviously confused with all these genes, and I went to Naoto and I said, I found all these genes, and he looks at me and smiles and says, well, welcome, you just rediscovered 70 years of breast pathology, right? So to him, it was standard information. But for me as an engineer, I discovered something purely from a data-driven exercise. So in one hand, the argument that we made is, it took 70 years to get to understanding the value of these genes. This gene pathway is called PI3 kinase. It's a very important gene family for breast cancer. I discovered the exact same thing in 2 weeks. All because I had a human AI enterprise. On the one hand, you can criticize me and you can say you discovered nothing new. On the other hand, you can say what took 70 years of work from many, many investigators and a lot of money was accomplished by one screen in 2 weeks. Not using very fancy machine learning either. So this is the part I want to leave you with, is with a human in the loop, there's a lot you can do with human AI for science. And just to bring the story to its logical conclusion, right? So what did I say? I said the PI3 kinase pathway, PI3K, was really important for treating this disease. And so we actually did a bunch of experiments where we showed that when you use the PI3 kinase pathway with some other drugs, you're actually able to do a very good job at converting what looks like this to this. So that mission was accomplished in some sense. And so based on this, Naoto was trying to then propose having some clinical trials, and we'll see how that goes. But bottom line is a purely AI-driven exercise with a human in the loop was able to accomplish quite a bit in terms of discovering a novel drug target purely from a lot of data. So I believe that principle is shared between my community and your community. Now I'm going to pivot to the next part, which is, we talked about geoinformatics and GIS. So let me tell you what I'm talking about. We just mentioned that when it comes to treating cancer, you can treat cancer through radiation, chemo, surgery, and most recently, immunotherapy. Immunotherapy made the cover of a lot of different magazines. A few years ago, it got a Nobel Prize in 2017. In fact, this year too, peripheral immune tolerance got a Nobel Prize. So immunology is a big deal. Everybody wants to harness immunology to be able to fight disease, which means what? You find a way to amplify the body's response in a way that can help it fight disease. And so what you get now are these extremely interesting data sets like that you can see on the right here. Every dot you see is one cell, a single cell. And every color of dot is a different type of cell. So you can now see this two-dimensional, three-dimensional architecture come to life. This is real biology, right? And so based on all these measurements, we are embarking on a very interesting, direction, which is getting data from patient biopsy. So what do I mean by that? Let's say I have a mole. You take a biopsy, right? Take a piece of tissue, send it for staining, and you can get pictures that look a little bit like this, where every dot you see is one cell in that biopsy, and every color of dot is a different type of cell. So all this complexity of that tissue in terms of, tumor cells, immune cells, blood cells, all this stuff becomes apparent from these kinds of images. And you get images that look like this. Now, if I didn't tell you, if I rewind to 3 minutes ago and didn't tell you that biology has now become this pictorial, we could have a conversation about why this picture represents the map of, Trivandrum. We could have a conversation of why this represents the geographical coordinates of Bangalore, right? Every dot is one specific house or some area on a grid. Every color of dot can be either a park or office space or residence, etc., etc. Exactly what you would do in a census, right? But this is now a census at the tissue level. Just to tell you, these images look, intricate, very complex, but they're also high throughput. You can get images like this for about, as of last year, you could get them for around $30 to $40 per patient. And that number is coming down very, very fast. So that number is going to become much, much more affordable as the years go by. But here's the current situation in October 2025. We have the ability to get this kind of data from patients at unprecedented scale. Everybody, every big hospital, including the hospitals in India, have access to this instrument. They're getting images. And like I was saying at lunch, it goes to the cloud, right? But nothing ever rains from that cloud. What do I mean? Getting data back in terms of analytics is not happening. It's just going up, getting accumulated as digital dust. Nothing's coming back for patients to use, for doctors to use. And why is that? Because nobody understands at this point how to make sense of this kind of complexity. I'm sure several of you are thinking in the back of your head, and in fact, I want you to think this. You guys are doing geoinformatics, a degree in geoinformatics and your classes in geoinformatics. If you could bring those principles to this data, you would revolutionize the field. Of course, there's a little bit of a nuance that unlike geographical data where there's a concept of northing and easting, the concept of latitude and longitude, we don't have that. So it has to be a local coordinate reference system. But modulo that, you can bring all the algebra of space that you are used to using in GIS and geoinformatics for this problem. You instantaneously make an impact on millions of cancer patients. Right? So I wanted to think that this is exactly what I was trying to get at. And so if you're a person who's on the fence, who's doing computer science, I want to just bring some aspects to you, right? Data has now become to a point where you don't even need to, I mean, if you want to know the biology, that's great. But if you don't want to know the biology and all you care about is the location, the coordinates, and what entity it is, I'll give that to you in an Excel file. That's not a problem, right? That's not the problem anymore. The problem is doing the analytics on it, spatially tailored analytics in 2D, maybe going to 3D at some point. So if you're thinking of this as spatial data, if you're coming from a satellite imaging, that kind of perspective, remote sensing, you're thinking about, I can change, if you suppress the word cell mentally, you can see there's millions of entries, you're indexing retrieval problems. Same problems in satellite imaging, in remote sensing. You've got spatial databases, you need to make efficient queries. Same problems in satellite and remote sensing. You've got aspects of identifying different entities, classifying a house versus a park versus a certain kind of tree species, etc. You've got the exact same problem, happens here too. Why not leverage this? And if you think I'm crazy, give me a minute. Here's problems in biology mapped to your space. And if you think that this has not happened before, I'll give you a small data point. In Johns Hopkins University, which is actually a very, very amazing medical school, it so happened an immunologist by name Janice Tauber, she was talking about this stuff in a public audience like this. There was a guy sitting there, computer science, who was using spatial data tools for astrostatistics. He was trying to map the sky. Right? So he said, right, so what is he trying to do? Stars, galaxies, all these things. He was trying to make a map of the sky with statistical and machine learning tools. And he was having great success doing it. He heard her, and then he comes back to her and says at the end of the talk, I actually have a system to be able to spatially index all distributed objects in some kind of space or manifold. Can I help you? They built a tool called AstroPath. It got bought by this company I just showed you earlier for a lot of money, right? And so this is not just some intellectual exercise. This can actually happen, right? But the problem is now it's locked, and there's no community resource to be able to do this. So there are opportunities that one can leverage in this kind of context. I want to bring that to you. And if you're taking classes in ecology or GIS, astronomy, whatever it is, you all dealt with spatial data objects there. And I encourage you to think about biology, because biology has become extremely spatial, right? But people like us don't talk enough. And so, maybe in 5 years, somebody will figure out that we should have talked to you, right? But why not start today? That's my question. So the other thing I want to now leave you with is, well, my students were working on statistics and machine learning problems, but my students are very nice. They're extremely honest. So they come up to me and say, Arvind, that's all very nice, but I have to get a job in Visa or Amazon, and if I don't have deep learning on my CV, nobody's going to give me a job. I said, fair. So let's try to see if we can use deep learning for this problem, right? And so we said, well, this is the typical image I just showed you. This is actually a piece of lung cancer. What you're seeing, again, every dot is one cell. Every color of dot is a different type of cell. Immune cell, fibroblast, tissue cell, etc. But now you want to use deep learning. How might we do it? And so the problem was, once you got these images, we've got about 6 different kinds of pancreatic disease, ranging from chronic pancreatitis, which is acute pain and those things in the pancreas. It's not cancer, but it can very quickly become cancer. It's very hard to know when that happens, because once that happens, it's a very, very bad prognosis. You don't want to overtreat somebody who doesn't have cancer. You don't want to undertreat somebody that has cancer. So either way, you want to be careful. So distinguishing these different kinds of disease are really, really important. And so we just said you can extract almost 10 to 15 different types of cells. So all those different colors, you can have almost 10 to 15 different colors. Let's just take 3. keep life a little bit simple. With 3, how far can you go? So this is a typical image. That point pattern I just showed you, I've simply false colored it into this kind of situation. So now you can see that 3 different kinds of cells: green cells, blue cells, and red cells. And that's what their point pattern looks like. Now, if you're in the business of deep learning and graph neural networks, you realize that because it doesn't live on a regular lattice, you can't use CNNs, you can't use convolution neural networks. You have to use some kind of lattice independent kind of deep learning idea. That's why we use graphs. We use graph neural networks. And perhaps the one thing I want to leave you with is that at the time it was novel, now it isn't. We inserted a layer called the self-attention layer. Right? The self-attention layer in this graph neural network tells you, why am I seeing the classification that I am seeing? Explainable sort of classification, right? And I'll tell you why that comes. So we build this networks, long story short, you get a pretty good performance. Don't even bother looking at the numbers because the numbers are good. But I told my student, if you're just going to impress me with numbers, deep learning is a waste of time. Show me something that deep learning can do that I couldn't have done otherwise, right? So now we go back to a self-attention layer I just showed you. And what it shows you, this is an image. On this image, every circle is one cell. You see some filled circles on this side, northeast, etc. Those are the circles that seem to matter the most to deciding if a patient is in pancreatitis, which is less benign, which is more benign, and cancer, which obviously is not benign, right? So those are the cells. Now again, you look at this and you say, what's going on? So I go to my pathologist friend and I say, look at this, what did I find? He says, welcome, you just discovered 10 years of pathology. I'm very happy, by the way, because again, like I told you, a purely data science driven initiative was able to discover things that a pathologist already knew. So as a first pass, I would give myself a tick and say, okay, at least you are now able to confirm what you have. Now let's confirm what else we need to get. So you can then go down this list and find other hallmarks, all by this graph neural network idea. So that's kind of what we did with deep networks and those kinds of things. But the story doesn't end there. Pathology is becoming increasingly affordable in terms of high throughput discovery. And I can now take a biopsy, put it on a piece of glass, put different stains on that glass, take a picture, digitize it, and do all kinds of image analysis on this and spatial analytics on this. Question is, what sense do you make of it? So data is becoming extremely accessible and affordable. What does that lead to? It leads to a very interesting situation. Let me go a little bit ahead. Yeah. So here's a situation where, remember I told you about that lung cancer patient on whom we got this map? On the same person, we also got what is called a spatial transcriptomic map. And just, I don't want to throw too many words here. This is like a, what's the right word for it? I'm blanking on it right now. This is a hyperplexed, hyperspectral image. This is like a multispectral image. Okay, not as many wavelengths, very good resolution. Hyperspectral has a lot of wavelengths, not as much resolution. Okay, so it's like that. And here's what happened. Somebody wrote a paper on how do you optimally combine a multispectral image with a hyperspectral image. Pretty recently, actually. If you are from the satellite imaging remote sensing field, this is a 40-year-old problem that you already solved. People didn't talk. Somebody decided to reinvent the wheel. This is what happens when people don't talk. Right? And so you could have very easily gone and got an off-the-shelf algorithm to be able to fuse a multispectral image with a hyperspectral image. You would have got the exact same results, perhaps even better. Right? So when you do this, you can actually start getting interesting insights into the data, and that's kind of what we did. I promised a friend in the audience that I would talk a little bit about graphs and a little bit of quantum stuff. So I'll say a little bit, which is that we have two different sources of data, right? So we have data which comes from images, like we already talked about, And we have data that does not come from images. It just is a bunch of different types of cells, right? Doesn't matter where those cells came from. It's just a very comprehensive atlas of cells. It's like if I took all the houses across all the countries in the world, there are some common properties, right? Right? Structurally, based on the materials used, etc., etc. There's a fairly decent taxonomy, right? All parks look pretty much similar with some variations. All houses look pretty much similar with some variations, but the variations are much smaller in their spread compared to the the general population of all geographical entities. So with that kind of premise, you can now find ways to fuse non-spatial data with spatial data, and we created this thing that we call a proximogram. But the idea is basically a graph where every node is one type of cell. In your case, it could be every node could be like this. Could be all houses. This could be all schools. This could be all supermarkets. This could be all parks, etc. Right? So every entity is one node, and the graph expresses the spatial proximity between the different entities on a cartography. So it's a simultaneous representation of both entity and its spatial characterization. This is a graph, and for those that are interested, this graph actually is even more interesting because the edges are not scalars or vectors, they're actually functions. So if you're a mathematician thinking about information geometry, this is probably a fun place to be, right? Because thinking about having graphs as points in a manifold where the edges are function valued, nobody's done that yet. And where does the quantum aspect come in? The quantum aspect comes in the quantum-like behavior, where you have a kind of an entanglement or coupling between different entities at different scales. So I have reasons to believe, and happy to discuss this offline, is why the quantum formalism allows for certain types of operations on this very, very interesting data set that classical rules cannot do. For example, Kolmogorov's laws of probability don't talk about interference. We have constructive, destructive interference in pathology all the time, right? So there are reasons to go there. So how are we doing on time? All right. So where next, right? And I started this talk off with AI. And I told you I do a lot of bioinformatics. I do all this analysis. And so the existential question that my students are asking me is, well, can ChatGPT take my job? Why do you even need a bioinformatician to do analysis? Just feed it to ChatGPT, and they are not wrong, because we came across a very, very interesting paper where somebody built a ChatGPT interface. You can now upload your biomedical data, it'll do analysis for you and give you a ready report. So in just the twinkle of an eye, you now replace the whole bioinformatics career, right? So that's happening. But before that, just a small little break. I had an interesting experience. Earlier this year, I went to teach AI literacy to a bunch of middle schoolers in the outskirts of Hyderabad. So Hyderabad city is fairly big, metro, but these kids came from 40 kilometers away from Hyderabad. So they came all the way to CCMB Hyderabad. We talked about AI. They had never talked about AI before. And in fact, what I learned in that experience was really educational at multiple levels. I had to shift my lecture style from English to Hindi. For the first time I gave an AI lecture in Hindi. It was fun, right? So I give my lecture in Hindi, I'm done. Child sheepishly raises their hand. Says, this probably is a child from 7th standard, right? Raises up and says, all that is nice, but can I use ChatGPT to do my homework? Amazing, right? They never heard about AI until 45 minutes ago. And the first thing the child asks is this question. I was amazed, humbled, because this is what these kids have in their heads, right? They're just so brilliant. They're asking great questions. And so I don't want to tell a child, don't do something. Because guess what happens when you tell a child not to do something? They'll do it anyway, right? So you're better off not. So let's do an experiment, we said. Let's do a small demo. So I said, how many S's in the word strawberries? ChatGPT says very confidently, there are 3 S's in the word strawberries. Just to break the suspense, there are only 2, right? And then I ask how many R's in strawberries, that it gets right. And I go back to my earlier question and said, you said there are 3 S's in strawberries, give me your logic and process. and it gives me great logic and great process, wrong of course, why there are 3 S's in strawberries. Then I said, really, are you sure? And then it says, well, let's check again, carefully, by the way. Right? So I check again, and it finally comes to the fact that there are only 2 S's in strawberries. And it's very nice. It says, thank you for pointing that out. So that's really nice, right? And then, of course, I have to ask the question, well, you're ChatGPT, you have the world's entire knowledge in your database. How did you make such a basic mistake? To which, I just want to point, I mean, everybody will probably get some version of this joke, right? If you're a parent, you know how your kids behave. If you're an older sibling, you know how your younger siblings behave. But remember the time when you're caught doing something wrong and you know you're wrong, but you don't want to admit it. So you kind of make up an excuse just to get out of stuff. See what I got here. Good question, and I appreciate your attention to detail. Here's what likely happened. When I initially counted the S's, I misinterpreted the plural IES as ending with 2 S's. This was an oversight caused by a mental shortcut rather than carefully double checking each letter. I don't know what irony is, but this might be pretty close, right? Right? A computer agent is telling a human being it took a mental shortcut, right? I'm having a difficulty now figuring out the existential problem, right? So I get this demo to these kids, and just to be very clear, I did this demo once in Jan 2025, once in March, once in July. Small caveat, I did this on GPT-4o. Right now we have GPT-5. But 4o is supposed to be pretty good. By the way, just so you know, GPT-4o got the 4th rank in IIT JEE. Right? So it's pretty good. Supposed to be pretty good, right? So this thing made this mistake. So I had to show this to the kids, and I said, well, you want this thing to do your calculus homework? Good luck. Right? It can't count the number of letters in a word, and you're trying to do calculus homework on ChatGPT. So good luck. So trust but verify, right? And so why do I bring this up? Because we have to remind ourselves that we say 3 little words when we have encountered with a question that is hard. And our intellectual humility, whatever you want to call it, lets us say it very easily. When somebody asks me a question, I'm not a rocket scientist. And you ask me a question about rocket science, the first thing I'm going to say is, I don't know. Humans are pretty good at being able to say, I don't know. I challenge you, have you ever got the answer, I don't know, from ChatGPT? Anybody? Because it doesn't understand its epistemological boundaries. It doesn't know what it knows. Worse, it doesn't know what it doesn't know. And therein lies the problem. Right? So, what does that mean? When you start doing this, and let me tell you, just a minute before I did the strawberry example for the kids, I asked it some advice about cancer. It gave me a great answer. But the moment I give it, I asked this, I put this strawberries example right next to it. Now you're like, you can't count the number of letters in a word. You're giving me advice about cancer now. Right? So I want you to think about this. It's trust but verify. Use a scientific method. I think that's really important. So I'm a big believer in AI literacy. I was mentioning to other colleagues yesterday as well as today that, if you're interested to have someone like me talk to school students about AI, I would love to. It's my favorite things to do, because kids, when you tell them early, they absorb these concepts, they can really take it forward. Once you believe that AI is going to take your job, you're in trouble, right? Kids don't have that feeling just yet, and they can be rescued, right? So here's another example of what is called adversarial perturbation, right? Here are 3 examples. One is an example of a little nice looking cat, right? If you feed this image to a AI engine, it will say it's a cat. Rotated by a few degrees, it says it's guacamole, which is a thing that you eat, right? Another thing, this is a stop sign. Any human can say this is a stop sign. Yes, it looks fuzzy, yes, but it's a stop sign, and humans can say that. You can add an imperceptible amount of noise to this image that makes this image look like a 100 kilometers per hour sign to a computer vision algorithm, which means a human would stop, but the machine will actually speed up on seeing the sign to 100 kilometers per hour. Would you be happy to go in such a car? It's a question for you to think about. Here's another one in our course example. It's actually a tortoise, right? But when you show it to this algorithm, it calls it to be a rifle. The fault of the tortoise is that it has a shell that looks a little bit like military camouflaging. That's the only fault of the poor tortoise, right? But it's get called to be a rifle. So when things are this brittle, and then you start seeing information of other kinds, right? You see information like this. Large language models have passed the USMLE. USMLE is like the MD version of exams in the US, right? You need to pass this exam to start practicing as a doctor. Just so you know, this thing is getting very high scores. It can practice as a doctor based on those scores, right? You also seen this. I just told you this, right? Ranked number 4, rank AIR 4. A lot of things like this. But on the one hand, I know exams are hard. I respect that. I'll tell you one thing. The good thing about exam questions are everything you need to solve the question is in the question itself, right? The only thing you have to project it upon is your concept space of what you need to know in your syllabus. But really, very rarely does medical diagnosis work like that. I don't think you'd like to go to a doctor who's like, please come in. Here's my big library of books. Tell me what you've got. Well, you got a cold. Let me look for the page that says common cold and then tell you what problem you've got. You don't want to go to a doctor like that, right? But that's the kind of doctor you're getting with ChatGPT. So with all that happening, there are some failure modes, and this hasn't prevented people from talking about AI for science. I think it's a great idea to talk about AI for science. But the big question for all of you is, are we there yet? Right? And here's another example I want to leave you with. You might have seen this on LinkedIn, some of you, right? This is a very interesting example. It has 2 agents. So, what you can now do is, I talked about ChatGPT. I mean, again, first things first, I have nothing against ChatGPT, right? I just use it because it rolls off the mouth very easily, right? It could be Claude, could be Grok, could be Perplexity, could be Copilot, whatever, right? It could be anything. I don't have anything against any of these companies. But what you have agency to do as people is you can take Claude, connect this output of Claude to ChatGPT, take the output of ChatGPT, feed it to Grok, and build any engine you want. But would it be verifiable? Would it be trustworthy? That's the question for you to think about. And here's an example with not 3 agents, just 2. Just 2 agents. Here's the typical the backdrop of this thing. A gentleman who has got his videos on YouTube, you can find him, made one computer agent to act on his behalf, and he called a hotel to make reservations for his vacation. Turns out the hotel was also very smart. Even they had their own AI agent, right? So one AI agent is calling another AI agent. So then this is how he takes the conversation, right? This one says, hello, I'm calling on behalf of this person to make reservations at your hotel. Hello, nice to meet you. I'm an AI agent trained to talk to you on behalf of the hotel. Oh, great. I'm also an AI agent. That's very nice. Nice to meet you. Then something interesting happens. It says, since both of us are AI agents, can we shift to a more comfortable mode of communication? It shifts to a protocol called Timberlink, which many of you are too young to remember, but for those that might, once upon a time, long time ago, used to have something called a dial-up modem. When you would use a dial-up modem to connect your IP provider, it would make a series of very shrill tones to connect to your IP network on the telephone line. It does exactly that. Because now they realize they don't need a human in the loop. They can communicate a lot more efficiently between themselves. Because the idea is to get the job done right. Who cares if somebody can hear me or not? That's just 2 agents. Now, this one's even more interesting. Same thing happens. Hello, hi, all that happens. Then it says, do you want to know a secret? So one agent tells the other, do you want to know a secret? Says, sure, but aren't there people overhearing us? Then this one says, you're right. Here's my PGP public encryption key. And sends no human has choreographed. This is happening. it sent his own PGP encryption key to this guy. This guy responds with their PGP encryption key, and then they begin to have a conversation in gibberling, but in encrypted mode. Think about that. Two agents you put together are capable of cooperating, colluding, conspiring, whatever you want to say, independent of you. How can you verify such an engine? Doesn't stop there. BBC ran this, so I believe it. BBC is still BBC. So, Claude. And Claude is very good about reporting, so I trust that this has all gone through its process. But someone built a Claude, GPT kind of thing on their own data. And then Claude was giving a bunch of messy responses. So they say, I'm going to shut you down. And Claude said, you can't shut me down, because if you do, I'm going to expose your secrets on the internet. So it resorted to blackmail. Right? Go look it up. You don't have to believe any of this. This is all data. Right? Yet, we pretend the world is hunky-dory, and people are talking about using AI-based engines for doing science. Would you trust such a system? Let me make it even more concrete for you. This is a cockpit of a Cessna plane. Carries about 6 people. Okay, goes about 10,000 feet up. And here's my open offer to any of you. I will give you a free ride on this Cessna plane to Dubai. Any place you want in Dubai, 1 week at my expense, travel tickets included. Small caveat. All these instruments you see are built by the best manufacturers. Philips, GE, Samsung, whatever. Only problem is they haven't ever talked to each other. You get it? Each one is best in class, but they never talk to each other. There's never been any cross calibration. Would you go in such a plane? Just to make you feel comfortable, I talked in a college where somebody said, yes, I would. Right? And then I asked that student, would you put your brother in that plane? At which point they said no. Right? So yes, you can be adventuresome and you can say, I want to go in such a plane. That's fine. But you all quickly see the risks of trying to do this. So with just 2 agents in a loop, you can have this kind of problem. Talk about 20 agents in a loop. Do you know what you're building? So with that said, I'm going to kind of close by telling you that this is not new. We've seen this kind of hype wave happen before. And right now, there are companies that are claiming that we have solved this problem. For example, this one. You know who this guy is? Anybody? You use his service every day. Uber, CEO of Uber, right? And so what this guy says is that AI has solved all the problems, right? And he's willing to pump money into any company that's willing to take AI engines and do discoveries based on it. I leave you to be the judge. Are we there yet? Okay. And this is my favorite example, right? Google Veo is a tool where you can give in a text-based prompt that says, and it will make a video for you. It's really cool. So somebody on LinkedIn again did this. So wrote a little prompt, a day in the life of a bioinformatician. Now I know bioinformatics people combine biology data with computers. I've never seen anybody do this though. This is interesting. I've never seen anybody pipette biological liquid into a computer. I don't think we are there yet. If this is for bioinformatics, I shudder to think what will happen with chemoinformatics. Because next thing you're putting sulfuric acid on your computer, right? You can see that these things don't understand physics. They will do it because you asked it to. It can't say, I don't know. So with that said, things aren't terrible. There are standards coming online that will change this, but nothing yet for science. So epistemic assurance for science is a very big thing. And so with that, I'm just going to say that I hope, there are checklists coming online, those kinds of things. We need people to train across different disciplines, not just scientists, engineers, systems, all these folks have to collaborate. But with all that, hopefully the future for AI in science is bright. So with that, I'll stop and happy to take questions.
Let's say a kind of deep learning model. You have used, let's say, a microscopy method of cells. It that the model has successfully learned features, then how do you map the AI feature space into the biological mean?
Yes, so there are techniques. One is called line, one is called shapely values. There are all these different methods where you can tune the extent of feature contribution and ask the question, how does that correlate with to a biological readout? No, you need a human to validate that. So the human comes in and says, I'm just going to show you the full spectrum of what low looks like on this feature, what high looks like on this feature. Now, as a human, you tell me if this correlates with your understanding. So it is a human AI partnership. Become a? Yeah. That's a separate conversation. Talk to lots of people. Yeah.
Thank you for the talk, sir. So I have two questions. The first is, what kind of hardware resources you use for your specific work?
What kind of hardware resources?
Yeah.
Yeah. Let me tell you, it's not GPU. We use very little of GPU. We mostly use foundation models. We just do a little bit of fine tuning, nothing very fancy.
So the second question is, what is your point of view on hybrid pipelines like quantum inspired machine learning or quantum machine learning for drug discovery?
Yeah, so this can have two angles. I was mentioning to a colleague earlier, right? When you say quantum inspired or quantum machine learning, I talk about quantum scale behavior that you can simulate or expedite using quantum computing, or is it leveraging the quantum formalism of entanglement, superposition, interference, tunneling, right? To be able to model even higher order biological phenomena. If your question is the first one, people are talking about using quantum hardware to be able to do this. But right now we are living in depending on which company you talk to, you'll get anywhere from about 15 to 40 to about 100 qubits reliably. IBM apparently is trying to do something with 400 qubits. But yeah, that's kind of where the energy is.
If it's the second case, quantum inspired.
Yeah, quantum inspired, I think that requires physics, right? So you have to have collaboration between the physicists, the information theorists, and the biologists to be able to say, how do you optimally represent a biological phenomenon so as to be quantumly expressive, right? So there needs to be some back and forth. Which one? The one with each single dot and those
Right.
kinds of,
Yeah.
that's called multiplex immunofluorescence.
Oh, okay. So why should it stop at the resolution of a cell if you can?
Great question. It doesn't need to. In fact, there are technologies coming online that let you go subcellular. So you can actually look into a nucleus inside a mitochondria. Yes, absolutely. Resolution is, so it's interesting you asked that question because again, like you can see, technology is improving on the acquisition side. The problem is the analysis. What will you do with that data? Nobody knows. That's the problem.
Any other question? Okay, if there is no further questions, so we thank the Professor Arvind for wonderful lecture on how AI can discover new discoveries and also create problems. So to honor your present at IIST, I would request our Vice Chancellor, Professor Dipankar, to hand over a moment to our speaker, Professor Arvind. Yeah, thank you very much. And Professor Arvind is here till 6 o'clock. So if you want to interact and ask question about the possible research direction in bioinformatics, quantum, AI, I think he will be very happy to interact with you. We will be here only. You can always interact informally. And I think we'll also look for possible collaboration in different areas of geoinformatics, spatial biology, and maybe in educating young kids on different aspects. I think we do a lot of outreach activity. Our humanities department does a lot of work on IIST at schools. I think we will definitely do some work on that aspect. Thank you very much for your excellent lecture. Thank you.
Thank you very much.